and some of the research that some of the research I'm going to talk about today is unpublished. So take everything I say with a grain of salt. Okay, so I think the reason why we're all here is because we have these new spatial measurements that are transforming our understanding of living systems. The way in which I like to view these methods in relation to the previous generation is that the previous generation of imaging methods, sort of gave you 3-color images of what biological samples look like. And so you can look at, some notions of, texture and spatial orientation, whatnot. Modern methods provide you now with n-color images, n being a number between a dozen and several thousand, where each quote-unquote color corresponds to some biologically meaningful variable. So, for example, you might look at the location and abundance of dozens of different protein species at the same time, or the location abundance of thousands of different RNA transcripts at the same time. And the nature of the data that can now be generated is really night and day compared to what could be what was previously possible. Classic imaging methods here on your left, the more modern data shown on your right. And if you get a headache just looking at what's on the right, it's almost kind of by design. I intentionally put too much information, too many colors on this image, just to try to relay some notion of its information density. And really what my lab has been focusing on the last few years is building these AI methods, AI systems that can render this modern data interpretable. So rather than looking at this image and getting a headache, instead you can see, okay, I see where the cells are, the relevant cell types, spatial patterns and whatnot, and have the AI methods really make that latent information accessible to you in a way that the human brain can understand. If we and this broader community of AI methods developers is successful, then there's sort of a whole range of biological discovery that's literally right in front of us, from understanding the spatial variation of gene expression to the spatial organization of different parts lists, looking at organization across different scales, going from the cellular to neighborhoods, communities and whatnot. And then, more practically, thinking about, okay, what are the different biomarkers, both parts list and spatial, that are linked to different disease states, and then through that biomarker creation process, understanding, okay, which gene targets are more relevant or not for disease progression. All of this is sort of the world that we're all trying to build, and I think it's only really through operationalizing this modern spatial data that we'll be able to get there. Okay, and so that leads to these AI methods, really deep learning, and if there's one take home I'd like you to leave with, it's that this deep learning revolution is data driven. These deep learning methods can do remarkable things on these spatial data and imaging data, but they can't do these things without labels. They have to learn from labeled data how to perform these particular tasks. And so if you want models that could say identify cells, identify cell types, really at the heart of these systems that can do this is large amounts of labeled data that are representative of everything that you might want your model to be able to perform well on, where that insight that the model is supposed to extract has been provided to it in the form of this labeled data. And as exciting as the unsupervised or self-supervised deep learning space is, I think the reality is that there's no real revolution without data and without labels. Okay, and so just to give you a flavor of the types of systems we're trying to build, I have this toy example of sort of the flavor of AI system that my lab really has really been trying to build. And here it's just this toy, this classic image of an immune cell, in a sea of red blood cells chasing a bacterial cell, and finally it finds a bacterial cell, engulfs and eats it. Really what we'd like to have is an AI system that can interpret this raw imaging data and produce a narrative, just like I described, with entity level resolution, right? So it should understand that it's this particular immune cell chasing this particular bacterial cell, finally engulfing it and eating it. And so you can go through a thought exercise of, what sort of AI modules would you need to build such a system? The very first module is, an AI that understands the notion of what's a cell, right? It should understand, that this is a cell, this wiggly massive pixel is a cell, these floating black dots are cells, and whatnot. And once it understands where the cells are, it should understand some temporal consistency, i.e., that as you go across this movie, that these pixels are sort of referring to the same cell. Then it should also be able to extract information, location, appearance, and whatnot. But that's not all of this. And so there's this layer of semantic information on top that you want this AI system to also be able to understand, right? It should understand, that these cells in particular, red blood cells, that this is an immune cell type called a neutrophil. These wiggling black dots are bacterial cells. And then you also wanted to understand the relevant interactions between the different cell types and how those interactions change the cell state, right? So it should understand that at the beginning of this movie, there's this interaction between this neutrophil, this bacterial cell called chemotaxis, and towards the end of this movie, this switches to phagocytosis. And once this phagocytosis event happens, there's a change in state of this bacterial cell from being alive to being dead. And already you can sort of see these AI modules, these computational primitives that you need for the system to be operational, right? You need to understand, where the cells, so cell segmentation, you need to be able to follow them over time, cell tracking. You need to be able to identify cell types, so cell type detection, and then this cell-to-cell interaction detection as well. And so really what these computational primitives do, if you have all of them working together in the system, is they let you convert this raw imaging data into this more interpretable format, a spatial temporal graph. Okay, and so the rest of what I have to say sort of centers around our efforts to build these computational primitives. The very first one I'll tell you about our efforts to do single cell analysis, just generally across all flavors of imaging data. It's a very common challenge for cellular imaging experiments. So this is true whether you're doing live cell imaging. So on the left is a movie collected in our lab, live cell imaging of a fluorescent reporter for the AMP kinase signaling pathway. And really what you'd want to be able to do to quantify these flavors of movies is, identify where all the cells are, track them over time, and then query each compartment, nuclear cytoplasm, how bright are you? And then this lets you pull out these traces of signaling activity. Also very important for analyzing modern spatial imaging data of human tissues. And so I alluded to before, there are these new measurement platforms that let you stain either dozens of different protein species at the same time or up to thousands of different RNA species at the same time. Really gorgeous data. And so this is paper taken from one of Mike Angelo's papers where they're profiling forwarded patients who had triple negative breast cancer to look at tumor immune interactions. And you can see, for each of these different 4 patients, you get a different image for each of the different protein markers present in their antibody panel. And so double-stranded DNA highlights, where the cell nuclei are. CD45 highlights the immune cell populations. P53 reports on the state of cancer cells and whatnot. Really gorgeous, really rich data, but to make it interpretable, one of the very first things that everybody does is cell segmentation. And concretely what that means, you have to go into structural images like this. So this is stained the double-stranded DNA, highlighting the cell nuclei, and you have to identify, okay, cell number 1, cell number 2, cell number 3, so on and so forth. If you're able to do that, then you can sort of quantify the amount of each marker each cell. This lets you identify things like cell types, cell states, and if you have cell neighborhoods, and if you have outcomes information, you can sort of link this higher order information to patient outcomes. Okay, and so what we've set out to do is sort of build the underlying data sets that you need to power general purpose cell segmentation algorithms, sort of our first computational primitive. And so what this entailed is, sort of collecting representative data across all of the different imaging platforms and tissue types being imaged for tissues, and then also collecting representative movies for live cell imaging data. And this includes both label-free modalities like phase microscopy and also fluorescent modalities as well. And so I just show this image just to give you a sense of, okay, what are the challenges associated with labeling these data once you have it. And so there's a challenge on the difficulty of each data type. So for the tissue imaging data, this is largely represented by cell morphology, which varies quite significantly across different flavors of tissues. In some tissues, cells are tightly packed, in others, cells are more sparse, and then also even within each tissue, the range of cell morphology you see is quite significant. And then there are also challenges as far as signal to noise characteristics across different instrument platforms, and then also artifacts that come about from doing 2-dimensional imaging of fundamentally 3-dimensional objects. for the live cell imaging data, the real challenge here is time. And so this is ensuring both that the labels that you have are temporally consistent, and then also labeling the full distribution of the dynamic events like cell division that happen in these movies. Okay, and so the second problem, in addition to the challenge of each data set, is scale. And so what this means is that there's just simply too much data for humans to ever label on their own. And the individual data sets are also too hard, for the AI methods to learn without labels. And so the question then is, what is one to do? And so the solution that we've been pursuing for the last few years is, image labeling at scale through human AI collaboration. If there's too much data for the humans to do, and if the data is too hard for the AIs to do, what about having the humans and the AIs work together to collaboratively label these data sets? And in that world, really the challenge then is sort of building the software systems that you need to enable this sort of collaboration. And so we've built, one such a software system called Deep Cell Label, which is shown here on the right. And you can see a human annotator going through a labeling task. Here they're highlighting cell divisions, but we build variants of Deep Cell Label for all the different types of labeling tasks that one might encounter, from labeling cells, labeling cell types, labeling dynamic movies, etc. And I just want to highlight that the key thing that enables scalable labeling is having humans correct the mistakes of these AI models. And so when you're starting with when you're labeling a data set, rather than having humans create everything from scratch, which oftentimes is something what you have to do at the very beginning, the more scalable form of labeling is having humans correct the mistakes of these AI models, and then having that corrected data then feedback, expand your training data set, and let you train bigger, more powerful, more accurate models. Okay, and so with that in mind, with a software tool and this labeling, this human labeling framework, we sort of set out to label, this large collection of imaging data that we've compiled. So for the live cell imaging data, we've recently put together a data set that's described in a preprint that we pushed out I want to say a couple months ago, describing how we can take this human loop framework, extend it to dynamic data sets, and then use the resulting data to train deep learning models, not just for segmentation, but also for cell tracking. And not surprisingly, because the models are powered by larger data sets, they end up being more accurate than what was previously available. We've also done the same for a large collection of tissue imaging data. And so about a couple of years ago now, we published a paper describing the construction of this data set called TissueNet, collection of over 1 million paired nuclear and whole cell labels across 12 different, across a collection of different tissue imaging platforms, tissue types, and disease states. And the really nice thing is that because because we have this expansive data set, we can train deep learning models on the whole data set. And because these models have now seen a wide variety of different cellular images, their ability to perform cell segmentation is substantially more general than the previous generation of models. And so here we see a collection of different tissue types. And here are the predictions produced by our AI method, Mesmer, that was trained on this diverse collection of tissue imaging data. And along with a visualization of accuracy, and then a accuracy score called the F1 score. And what you can see just visually, and then also by the scores, is that Mesmer is able to perform quite well across all the different data types. And that the key thing is that it generalizes. So this is the same model, same hyperparameter settings and whatnot, applied to all of these different use cases. So since Mesmer was published, we've been working on the problems that happen after cell segmentation for spatial proteomics. This is cell phenotyping, identifying positive markers and also cell types. And for spatial transcriptomics, this is spot phenotyping. So for identifying for each individual single molecule spot, identifying where they are, and then also what genes, collections of spots correspond to. And so we've been working on building AI methods for each of these different problems. For the cell phenotyping, I'm not going to belabor this point because you've all done really, really excellent work in this space, and I'm sure he's going to go into the details in his talk. But just suffice to say that, there are challenges still, even with accurate cell segmentation, that you're just never going to fully get away by having better segmentation. So segmentation errors are still a thing. But even in spite of that, there's noise, there's marker spillover, and then there's cellular projections that spill lineage marker across neighboring cells. And these are all artifacts that need to get taken care of. Once you're in this space of images, convolutional neural networks are appealing technology. And so, if one had a large amount of labeled imaging data, you could probably make some of these go away with a deep learning based classifier. But the biggest problem is that we see is generalization. There are a wide variety of different methods that are generating this flavor of data. Some of them based on imaging with light, some of them based on imaging with mass spectrometry. Each of them have their own nuances as far as signal to noise and autofluorescence characteristics. But then there's also challenges across different data sets that people are generating. And the reality is that, each paper that features one of these methods has, its own antibody panel that's speaking to the tissue of interest. And that makes it challenging for building the AI methods because each data set that you have is different, and these differences do matter. And so, as part of sort of data-driven methods development, what we've done is we sort of gone through the entire community literature, ingested all the publicly available spatial proteomics data along with metadata, and gone through an effort to try to integrate and harmonize these data. So segmenting with a common segmentation tool, Mesmer, getting a common marker panel, and then also a common set of cell types that can be identified with said markers. To the extent that they exist, pulling in existing cellular labels, and then also we have a labeling effort that's ongoing internally to sort of add labels from this harmonized cell type list to all of the different data sets we've put together. For the AI side, to have a method that generalizes across all these diverse data, really there are a few things that you need. A big one is normalization, but then on top of that, you need the models to have access to the full information content of the images, and you also need them to be aware in some way of what the marker panels are. So if a model's looking at an image of, say, CD45, it actually has some notion of what CD45 is, or the very least that CD45 is different from the other channels, CD8, CD4, ECAD here, and whatnot. And then also really important is access to these scalable forms of labeling. I will highlight that, as part of this work, there's just a lot of immunology and biology that has to get understood. And one of the students who's worked on it has built a language model powered assistant to help us, sort of make sense of all the different experimental data sets that we've been pulling in. And so what he's done is he's taken his immunology textbook, Janeway, and made it searchable using a language model. And so he's also combined that with the OMAPs produced by Kati Borner's HubMap group. And the result is, this sort of knowledge assistant that you can query for information about any particular marker as you're working on, sort of data set harmonization. And this is currently online. Okay, and so generalization, requires changes to deep learning model architecture, so that you actually have models that can access the full information content of these data, and that's really getting at being able to learn the correlations that exist across in signal across different markers, but then also can generalize across these different data sets. And so the approach that we've put together is sort of a hybrid between the vision models that are super popular, convolutional neural networks, and the language models, transformers. And so we're calling this sort of marker-wise attention, and this is I'll explain how it works in a second, but this is a computational feature that we believe is going to gives rise to the features you need for generalization. So the starting point is start with the tissue images, pass it through Mesmer, segment all the cells, and then crop out areas around each cell so you can see the cell, and then also some notion of its neighbors. And so, for each channel, so for example, CD45, CD8, you have these three images, where the cell, raw image of the signal, a normalized signal, and then images of the cell masks. This gets passed through a convolutional network, and then the end result is you get a vector feature that summarizes the content of these images. Then what you do is you take the marker, marker name. So for example, CD45, and then you pass it through an embedding model. And so this marker name actually gets converted into a vector and vector embedding as well. And so this model can be something that's learned on the fly. It can be a pre-existing one like ChatGPT. We've been exploring different ways of generating these marker embeddings. It's evaluating which ones actually lead to better generalization. But the core idea is the same: is that you just have this embedding model generate these feature vectors, and then you add them together. And so the end result is you now have this vector that's aware of what the image for that cell looks like, and then also semantically what that marker was. And so then you can take these, you can sort of generate these combined vectors for every marker image present in your data set. And then you can throw them into a transformer module that operates across the different markers. And this module effectively learns the correlations that are present across different markers. And so now you have these updated feature vectors that are more information rich because they've been boosted by this correlative information that was extracted. And then you can use those for all the downstream tasks. So marker positivity, cell type detection, et cetera, et cetera. For building out these data sets, we've adapted a deep cell label for doing human loop phenotyping. And the way in which this works is we sort of have a lightweight within browser classifier that operates on these embedding vectors that have been generated by models trained on available training data. And so what folks can do for labeling is just label some archetype examples of particular cell types in the field of view. Use that to train an iteration of this lightweight classifier. This classifier is uncertainty aware, so then you can use it to process more data, and then zoom in specifically on the example cells where the model's the most uncertain, right? Because you can sort of see that here, where the blue cells represent ones with model certain, the red are the ones where it's less certain, and you can sort of this iterative fashion zoom in on the uncertain ones, label some more cells, retrain a classifier, and continue the cycle until you're done with a particular data set. And I'd also highlight that we have a variant of this that we use for labeling marker positivity as well. And our hope is to have sort of this integrated data set where across all of the different spatial proteomics data, as well as the spatial proteomics data we've been ingesting from PubMap, we'll have both cell type labels and then also marker positivity labels as well. So this is work that's been ongoing. Preliminary models look like they went fairly well. So here is an example prediction of one of our marker positivity models. It's the raw image, ground truth, and the prediction. And you can see from the F1 score, that there's fairly good overlap between the two methods. Between the ground truth and the model predictions. And then we also have prototypes of this model that can for performing cell type prediction. And the key thing here is that these were models that were trained on on data sets with different marker panel. But you can see here that it's able to generalize and pull out these different cell types. And more examples, and the early benchmark that we've done is showing that this approach can work fairly well, and the confusion that you see is sort of confusion that you'd expect based on community experience. Okay, last thing I'll say is that we've been working on a method for spatial transcriptomics data. Here, the way these methods work is by doing sequential single molecule RNA FISH. And so you have a tissue, you have a panel of RNA FISH probes targeting different sets of genes. You'll do several rounds of stain, image, wash, stain, image, wash, stain, image, wash, and then you end up getting this series of images And then ultimately what you have to do is, spot detection, register each image, because, shifts happen across these different imaging rounds, do spot detection in each round, and then line them all up, see which detected spots are corresponding to the same, sort of the same molecule, and then, go from, those detections to which genes were present. And this is typically given by a code book that is designed before the experiment was done, telling you that which rounds should a given gene appear in. And so, one thing that's been missing from this space is just a general purpose spot detector. And there's good reason for that, which is that it's hard, and it's not hard from the AI side. It's actually fairly straightforward to write down an AI method that can do accuracy multiple spot detection. The real challenge is the data. There's just too much data for humans to ever label. A single experiment might quite literally contain millions and millions of spots. And so then, the question is, what is one to do? And so what one of my students, Emily Lauscher, has put together is a weekly supervised approach for spot detection, where, in this approach, you sort of take all of the, collect some representative spot data, get all of the classical spot detection methods, and then fine tune each method on each data set, and then do generative modeling to find consensus among each of these different methods. And then this generative modeling effectively denoises the predictions produced by each of these different classical methods. And so that denoised consensus label is what's then used for training a deep learning model for spot detection. It turns out that this works really well. So in fact that, you can sort of take the resulting model, combine it with a probabilistic gene decoder, and Mesmer, and then you have a end-to-end unified pipeline for spatial transcriptomics that works across all the different flavors of transcriptomics methods that exist. So from wordfish, seqfish, in situ sequencing, pooled optical screening and whatnot, this method works. And so we also released this as a preprint about a month ago, and it's currently under review. But the software is out now for people to try if there are folks in the audience who are doing spatial transcriptomics. And so that is it. I will stop talking, and then I will leave time for questions. Huge thanks to everybody in my lab who worked on all these projects. It goes without saying that I did very little of this myself. And what you've seen was work by several talented graduate students, Julie Wong, Rohit Dillip, Morgan Schwartz, and Emily Lauscher, as well as staff Ross Bernowski, Kevin Yu, Ada Atiz, and another software engineer, Shen Li. Have to give big thanks to collaborators, and then also different organizations for funding this